Broadcast is now starting. All attendees are in listen-only mode.
Welcome everyone to today's GastroPlus user group webinar on applying mechanistic absorption modeling and PBPK modeling to predict positive and negative food effects. My name is Philippos Kessisoglu and I'll be moderating this webinar, and it is my pleasure to introduce our two speakers, Dr. Tycho Heimbach from Novartis and Dr. Neil Parrott from Roche. Dr. Heimbach is a director in computational biopharmaceutics in the PK Sciences department at Novartis. He received his PhD in pharmaceutics in 2003 from University of Michigan, and he serves as the biopharmaceutics expert reviewer and makes recommendations and conducts predictions for formulations and physiologically based PK and PD strategies within the global PK sciences function. He has contributed to a priori PBPK modeling for more than 15 clinical trials and has authored more than 10 INDs. He has been strongly promoting the use of BCS and BDDCS in drug development for likely food effects within both Novartis as well as external scientific meetings and symposia. He's been a speaker at 25 national and international conferences, has authored or co-authored more than 30 peer-reviewed publications, 3 book chapters, and 1 patent. He is an AAPS volunteer for the last almost 10 years, and he currently serves as an AAPS ambassador. Dr. Neil Parrott is a distinguished scientist in the Modeling and Simulation Pharmaceutical Research Group at Roche Innovation Center in Basel. Neil works in preclinical modeling and simulation and specializes in physiologically based pharmacokinetic modeling and its application to Roche projects for early discovery to the market. He has a particular interest in the application of PBPK modeling for first-in-human PK predictions and to guide formulation development and pediatric drug development. He's also involved in external cross-industry consortia like the IQ Consortium, and has published over 50 papers and has given numerous presentations at international conferences. So with that, we'll move to the presentation, and Tycho will start us off.
Thank you, Philippos, for the very kind introduction. I'm going to start the broadcast. Today we're going to be covering physiologically based pharmacokinetic models for food effect predictions, as we are conducting them in drug development and sometimes in late drug discovery. You can see it's usually a team effort from a large number of people, and it usually requires successful integration of in vitro data, animal data, and in some cases existing human data to predict the effect of food on drug absorption. Our webinar is targeted both for experts but also for beginners of GastroPlus. So we will have a brief review of the food effects that you typically can see in drug development, and then we will have 3 case studies along with some conclusions. Physiologically based pharmacokinetic modeling or mechanistic absorption modeling, which is has become very popular with regulatory agencies and also industry, require a large number of different inputs. If we have the end game in mind, which is the prediction of both fasted and fed state, Then we can see that there is a number of different input parameters that are required. We can see there is drug-specific parameters that are specific to the drug molecule that you're developing. That includes ionization behavior and dissolution profile, along with biorelevant or clinically relevant solubility data, along with biorelevant permeability data, as determined from either KCO2 or MDCK, but in some cases also derived from animal in vivo data. Today we will not be having any examples that involves transporter involvement, but it can be included. On the population side and on the platform side of the software, we need to worry about physiologically based parameters. And today we will have an example where we need to think about how do we capture the pH in the intestinal milieu. Once you put together both the GI physiology parameters along with drug parameters, then you can have the foundation for the gas-to-plasma model. One of the components we also need to think about is the clearance and the volume of distribution. These are the disposition parameters that you typically get from an intravenous arm. And if you don't have intravenous data for your compound, which is common in early drug development, then you need to determine these clearance and volume parameters from preclinical data, or you can also use a whole body gas-to-plasma PBPK model. And this is just for as a reference from David Fleischer, who had originally written about food effects and how it relates to BCS classes. You can see a positive food effect usually means AUC or exposure is increased in presence of food. Negative food effect means the AUC is reduced. And today you will hear some examples where we are basically seeing no change. And I think everybody who works in biopharmaceutics and in food effect predictions should be familiar with the biopharmaceutics classification system, which has 4 classes. And today you will mainly hear about BCS class 1 and 2 drugs, where for BCS class 2 drugs in the presence of food, often an increase in the systemic availability, an increase in F extent is observed because of solubilizing bile acids that are released when you ingest the food. And for BCS class 1 drugs, often you see no change. Now, remember that food effects can be dose dependent, and they can also be formulation dependent. And as such, some class 2 drugs at lower doses could be more behaving like a class 1 where there's no change. And our webinar was partially inspired by some work that was originally discussed at the AAPS of 2017, and Neil will cover this a little bit more in detail, but by and large, there's still some skepticism among regulatory agencies whether food effects can be predicted reliably. In this particular data set, some critical comments about PBPK and the predictivity were made. So we wanted to better address these, and we put together from the gas-to-plasma user group a few colleagues who have experience in this area. And you can see this is a paper, Journal Pharmaceutical Science, which is currently in press. Christoph is the first author and Philippos is the corresponding author. So we put together case examples to demonstrate how and when food effects can be predicted with confidence. This brings us to our first case example, which we call here compound N. Compound N is a weak base with accurate solubility. You can see here 0.1 to about 0.4 milligram per ml, and you can see the solubility is also listed here in the fastest and feasible conditions. And the compound overall has high permeability in KCO2 and pampa assays, and there is no significant gastrointestinal transporter involvement. In preclinical species, the compound had complete absorption in radiolabeled study as determined by radioactivity. And in the other species also, when properly formulated, we had a high absorption, right? If you have a bioavailability of 100% in the dog, that means the absorption was 100%. Overall, there was no significant species disconnects for this drug. And there was also no issues around gastrointestinal instability or degradation. So for this drug, as we entered the drug development for the first in human studies, the question was, can we run compound N with a light meal, because the dosing regimen had been proposed to be BID twice a day, and patients and even healthy volunteers usually do not like to fast twice a day with a 4-hour fast. So the question was, can we run the studies with a light meal without compromising the findings of the study due to complicated absorption? And then number 2, can we predict and describe different prandial states? And there we used a GastroPlus, the bile acid biorelevant solubility data which we generated. And in terms of the simulation methodology, we used the ACAT model, and on the systemic side, this is consistent also with our publication, we used the compartmental PK using estimates from population PK. Most of the parameters that we used were actually defaults. For the ACAT model, we did modify the colonic water content to 2% in colon and 20% in the small intestine. The fasted state was by and large the default values within GastroPlus, and we had to create a light fat meal human physiology parameters or ACAT file. And I will show you how we did that. These days, we prefer to run virtual trials so that we can better predict the variability. And we will also show how we included variability around certain parameters in GastroPlus. The pH variability, for example, was set at 100% in fasted state in different sections of the gastrointestinal tract, which is different from the defaults. And for the clearance and for the volume, these were the percent CV were estimated from the population PK analysis. I mentioned on this slide here that we have to estimate the clearance and the volume for the compartmental PK. And so we did this using Wajima method and the recommended scaling methods. This is described in this particular paper here. Coming back to GastroPlus, most of you will be familiar with the fasted default for the human physiological fasted condition, and the fat default, which is the human physiological fat condition. In the middle, we have what I call LM, which is the light meal, which is 500 calories. You can see in the fasted state, the bile acid concentrations are less than 3 millimolar. And in the fat state, again, these are defaults, it's less than 15 millimolar. So we just scaled it based on the calories as an estimate, and we use this model for our prediction because the patients were consuming a light meal, were to consume a light meal. Then with the virtual trial, you can see here by and large we use the defaults. Again, we put a high percent CV on the stomach pH in different sections in the intestine. And what's cut off here is the percent CV on the clearance and the volume. Then you can see the data here. So this study was unusual because we wanted to run the study with food, with a light meal, to allow patients not to fast. So this is basically the breakfast condition. And you can see the observed data are here with the mean, and there's some variability around the data. And you can see these shows the observed mean in red and pink, and then we have the mean data here in black, and then the confidence around the mean is shown in green. And the probability contour plot was chosen to show the probability between 10% to 90%. So overall, the data is actually described reasonably well. The fasted data became available 3 years later, and we had done the prediction from the light meal data, and you can see that the model actually describes the data very well. Same as before, we have the 90% confidence interval around the mean, which is very well described, and also the variability of the population is well described with this particular virtual trial setup. This is helpful for informing project teams around what type of variability you might be getting. And then if you look at the individual numbers, again there in this publication, which is in press, you can see there is no food effect. Now remember that prediction of no food effect is important, because I would submit to you that there are studies that we're running which may not be needed, because we can use PBPK models, we can use GastroPlus to help us out with the predicting the outcome with confidence. Here we have a second case example, which is from my colleague Mark Lesney. And here the same question was, will we see a food effect at a given dose for compound E, which has high solubility, but permeability is only moderate. And solubility is actually adequate, but if you calculate the dose numbers are larger than 1 mathematically. So we had to do an assessment whether or not there would be a food effect. And as before, we use GastroPlus and we use the ACAT model. We use the biorelevant in vitro solubilities, and we use a pH solubility profile data. You can see the dose number here is actually relatively small, it's 8. But if we want to convince regulatory agencies, we have to do this type of assessment. And also we have to do this type of assessment sometimes to convince scientists and project teams within an organization. And so here we have now used the PK Plus, it's the module PK Plus within GastroPlus. We can fit the data with a 2-compartment model and then use the parameters for the systemic circulation, and we can describe the ACAT model for the pharmacokinetics in healthy volunteers. This again work was done by my colleague Mark Lesney, and then we use the top-down optimization to get the fit for this particular data set. And the beauty of physiologically based pharmacokinetics or mechanistic absorption modeling is that you can do the diagnostic plots. So we're showing here the dissolution is occurring at a faster rate relative to the overall absorption, which is dependent on permeability and or gastric emptying. And so you have basically dissolution safe space. It is very important to understand what is rate controlling systemic exposure. And if dissolution were not critical, then you would get more a plot like this. So in this case, again, we wanted to predict the food effect. This is a case example where we definitely dealing with high confidence, and maybe in the future such studies would not be needed. But what this was the question here is, when we have this particular capsule and we give it with food, what is going to be the outcome? So for those of you who know biopharmaceutics and who understand how Gastroplas works and the given solubility in the properties of the compound, you can imagine that there's not any food effect. So we did the what I call predicting the predictable. And you can see here's the fasted state, and here's the fed state. There are no surprises, but we're doing this work now to convince others that this can be done with confidence. And the clinical data is shown here, just for illustration purposes, where we had the fasted observed and the fasted simulated, and then the fed data does not show a food effect, and I could not show the real data, but there is no clinical food effect observed for this compound. Now, just briefly talking about the negative food effect. At Novartis, we routinely evaluate compounds where we're not sure if a bile acid complexation could occur, which could lead to a negative food effect, reduced exposure in the presence of food. So we use a modified KCO2 assay, which uses FASEF and FESIF conditions. And you can see the surfactant concentration here, where the surfactant concentration is higher on the gut side, the apical side, compared to the FASEF, and we measure basically the transport rate, just the permeability. And we have applied this routinely in our compound setting. This is an example which is now a little bit older, but it demonstrates the point very well that the biorelevant permeability can describe the negative food effect which we have observed, because we have an 8-fold lower permeability in the fed state due to complexation compared to the fasted state. So it is not only important that you understand the solubility, but also the permeability data when you're using those as inputs into a Gastroplas model. So in conclusion, physiologically based biopharmaceutics in PBPK is of significant regulatory interest. There remains to be a lot of science that needs to be done to convince industry and convince health authorities that the predictions can be done. So the examples that are shown here and also in the paper demonstrate how you can show a lack of a food effect, which is important because you do not want to run unnecessary studies if you can predict the outcome, especially what I call predict the predictable. So you have clinical data in one prandial state and you can predict the other outcome. So you can use fasted to predict fed, or as I showed you for the compound N, if you have a light meal breakfast, you can predict the fasted outcome. We also recommend that virtual trial simulation should be conducted by everybody who uses Gastroplas and who is part of the user group. I think this is my last slide, so I will basically hand it back over.
Okay, do you hear me and do you see my slides now?
You're good, Neil.
Oh, okay, great. So thank you very much, Taika. That was a really nice presentation. I think you did a really nice job of going into some of the mechanisms and details of the physiologically based modeling. In my half of the presentation, I'm going to focus on slightly different aspects. I'm going to look at the potential for impact with the regulators of this type of modeling. I'm going to look at how food effects impact the development of a drug and a little more detail on how much resource is currently going in that direction. I'm then going to share a Roche case study to, I think, illustrate some of those points made in the first part. And then lastly, I'm going to share some of the current ongoing activities and future directions whereby we can see potential improvement in the situation. So starting with the current regulatory guidance, so sponsors of orally administered drug products are required to conduct food effect bioavailability studies to support global filings of NDAs. And shown on this screen are the guidance documents relevant here. On the one side, you see from the FDA the guidance which dates from 2002. So you can imagine that with this guidance, which is now 16 years old, there's no consideration of the potential impact of PBPK absorption modeling within that guidance. On the other hand, the EMA guidance is more recent, dating from 2012, but likewise, there's no mention in the guidance of the potential for mechanistic absorption modeling and prediction of food effects. Both guidances are quite similar in what they mention. They mention that a food effect study is mandatory for immediate release formulations, and they both mention that it could be necessary in addition to perform additional food effect studies if formulations change, to explore the impact of different types of meal or impact of different timing of meals with respect to dose administration. And if we look at the timeline or the steps involved in drug development, we can see that quite typically more than one food effect study is performed. So quite often the first food effect study is done early in the clinical development in order to get a first impression of the effect of food and thus inform the clinical pharmacology studies that will take place in phase 1 and 2, and to get a first idea of how to dose the drug going forward. But of course that would be done with an early formulation, and formulations change during clinical development, and so it may be considered necessary to repeat a food effect study with subsequent formulation, and almost certainly by the time you come to the market, your formulation has changed, and it will usually be necessary to repeat a food effect study with the marketed formulation for labeling purposes. It's also worth to mention that in cases where you have a significant food effect, this can be considered a hindrance to the success of the molecule, and so there may be need to optimize formulation to reduce food effect, which in terms of clinical studies can be very time-consuming and costly and resource-intensive. So basically, the message is there's quite a lot of resource can be needed to understand and mediate food effects, and so if we can do something to optimize that, it's well worthwhile. And so that was something which we recognized at Roche quite some time ago. Indeed, in 2006, we published first paper on this topic. This was a paper where we looked at using PBPK modeling together with biorelevant solubility measurements. We looked at a set of BCS I and II molecules, which were in our in our development pipeline where we had already food effect data, and we showed with this retrospective analysis that we were in a position to make quite reasonable predictions for those food effects. Another paper came out in 2008, at that time in collaboration with colleagues from Simulation Plus, and I would say that from this time forward, we have been very systematically using GastroPlus for food effect predictions in our projects. And most recently, in 2016, we published another paper for our molecule alectinib. This was indeed a very challenging molecule with it was a BCS II molecule, but with extremely low aqueous solubility, and so the absorption modeling was quite challenging. Nonetheless, we still saw some benefit in use of the PBPK approach in this project, and I would refer you to the paper for more details on that work. However, for my case study today, I wanted to focus on a more, I would say, a more typical example, another BCS II molecule, but one which I think is representative of many other molecules in our pipeline where there could be significant impact of food effect modeling, and I think actually quite similar to the case study just shared by Tycho. This molecule has the work done on physiologically based PK modeling has been published in at least 2 papers, which you can see mentioned here. But the work that we did on food effect has not been published, and at least until the paper which is now in press with the colleagues as mentioned by Tycho. So to share with you the biopharmaceutical properties of this BCS II molecule, it's a neutral molecule with moderate lipophilicity. Human permeability was estimated initially preclinically using the PAMPA assay, and then later when we had entered into phase 1, more detailed studies were done with CACO2 and scaled to, as you can see, about 3-fold higher permeability. Solubility of this molecule was low, so only 5 microgram per mil in buffer, but the solubility was enhanced in the biorelevant media, so up to 17 in the fasted state media, and about 3-fold higher in the fed state media, so already raising a potential here for a food effect. The data mentioned here in blue, again, these were data obtained once we'd entered into phase 1 with the clinical formulation. And prior to entry into the clinic, we actually did all of the pharmacokinetic prediction for this molecule using GastroPlus, and we also at that time made a preliminary estimate of the food effect, and we found that, you know, we had an early estimate of the clinical dose would be around 13 milligram, and at that dose we did not expect any food effect. So then we went into the single ascending dose phase 1 study. We had quite some confidence in our PK prediction for this molecule because we had seen good in vitro to in vivo correlation in the animal species, and yeah, the molecule seemed to be well behaved in terms of its PK. And then applying the same approach based on human hepatocytes and physiological scaling, we predicted a low human clearance of 1 milliliter per minute per kilo. Volume was predicted as high at 3 liters per kilo. And the bioavailability was predicted also to be high, with absorption complete for doses of less than 80 milligram. Thereafter, a slight tail off due to incomplete absorption due to the low solubility. So at the bottom of the screen here, you see that our prediction was nicely confirmed by the first clinical data. On the one hand side, you see for the 50 milligram dose, the simulated line overlaid with the actual mean data at that dose in 6 healthy volunteers. And then the other side plot, you can see that over the full dose range in the single ascending dose study, we were well within twofold in terms of our predicted AUC. You can also see here with the black predicted bars that slight fall off in exposures above 80 milligram, as I mentioned. So, you know, we had now some clinical data, so we were in a position to refine the human model using those data and using some of the in vitro data which I mentioned in the first slide on this molecule. So you saw that we had a slightly higher solubility measured for the formulated material, and we had a slightly higher permeability based on the CACO2 data. And it was nice to see that when we included those data in the model, we got a much better description of the absorption phase using our PBPK model. And that was indeed the only modifications which we needed to make apart from a reduction in the colonic water volume, because we were seeing some late absorption leading to an over-prediction of the terminal phase exposures. And when we adjusted the water volume, that was eliminated. And so that's what we did at that time to refine the model. So at this time, the molecule was advancing, and it was decided that it would be needed to perform a clinical food effect study. I mentioned that our estimated dose was around 10 milligrams. However, there was uncertainty, as there always is, about the clinical dose. And to explore the food effect in the most stringent conditions here, we decided to carry out that food effect study at the highest anticipated clinical dose, which was 80 milligrams. So we used the PBPK model again to predict the food effect at 80 milligrams, and we predicted only a very modest increase in Cmax, around 1.3-fold, an even lesser increase in AUC with food. And you can see from the bottom row of this table that the clinical data nicely matched up to those predictions. So the model was really doing a nice job. And so then we proceeded, we continued to use PBPK modeling for this molecule. We had multiple clinical pharmacology studies where we were able to verify the performance of the model. And we came to phase 3, and at this time it was decided that a film-coated tablet would be most appropriate for the market formulation. At that time, we had refined the clinical dose to be 10 milligrams. We had also performed a relative BA study with those film-coated tablets compared to the phase 1 capsule, and we'd shown equivalence for those two dosage forms. you've seen that we had confidence that the model was capturing the absorption behavior because we'd had good match with clinical data and we'd predicted well the food effect at 80 milligrams. However, because at that time there was really no precedent, there was very little mention of food effect modeling. There was no precedent for acceptance by the regulators for PBPK modeling. And so again, we decided to repeat food effect study with the clinical formulation at 10 milligram. And this slide now shows that at 10 milligram we predicted absolutely no effect on AUC and a slight reduction in Cmax due to the delayed gastric emptying with food, which was perfectly confirmed by the clinical data. So absolutely no surprises here. So just summing up my case study, this was a BCS II molecule, but had very well behaved PK. The PBPK model based on in vitro data was well verified with multiple clinical studies. And so I hope you will agree with me that that second food effect study really added minimal value and could be waived with almost no risk at all. And based on my experience, I feel that molecules like this, and you've seen the examples from Tycho as well, this represents a good percentage of development compounds within BCS I and II classes where there could be great potential for optimizing clinical development and waiving of studies. Of course, we don't forget that other BCS II molecules can present more challenges, and I refer back to the other example which I mentioned of alectinib, which is certainly a much more challenging molecule with formulation effects which are more difficult. But I think we can recognize those molecules which are predictable from those which are not. Okay, so now I'd like to finish off this presentation with some slides on current confidence with PBPK modeling from industry and regulators and looking forward. So in 2015, I had the pleasure to be involved in an IQ working group with representatives of 10 other pharmaceutical companies, of which Tycho was one such representative. And we got together and we came up with a white paper, which we published and in which we outlined the impacts of PBPK in terms of different applications. And we also broke down those applications into the areas where we had high, moderate or low confidence. And I've pulled out the table concerning absorption, food effect and formulation modeling. And you can see that from our consensus opinion was that for BCS I and II drugs, we had high to moderate confidence in absorption modeling, including food effect predictions. So quite a positive level of confidence among the industry around predictions for this type of molecule. However, if we look at publications and presentations from the regulators at the same time, we see a mismatch. And so I've pulled out a couple of examples here. The first one is a paper from Christian Wagner and colleagues at the FDA. And you can see from this table that they highlighted that they had limited experience with food effect modeling and thus had low confidence in use of PBPK in that area. More recently, there was a webinar presented by Ping Zhao, representing FDA, and Adam Nordmark from the EMA. That was presented last year in September, and they both confirmed at that webinar that they had low confidence in absorption modeling, and they also made the statement that they felt this was not scientifically established yet. So yeah, clearly some discrepancy here between the level of confidence in the industry and in the regulators in this area. Wanted to mention also, and Tycho already mentioned this paper which came out also in 2017, an evaluation of food effect predictions using physiologically based PK modeling. Again, this was from Ping Zhao and colleagues at the FDA. And they were, not overall so convinced about the modeling here, making the statement there were large knowledge gaps in product API and physiology, which were hindering the ability to predict food effects. So the situation is quite clear, and so the question is, what do we need to do to increase that confidence among the regulators? And so, I'm speaking now for, I think, many colleagues in the industry, and in particular for the colleagues who worked on the current paper that's been highlighted. We felt that one thing that we can do is to address some of the concerns by establishing a consistent workflow and standardized inputs, which we all endorse. Of course, there are some key principles which should always be followed in order to have confidence in food effect predictions, and clearly the mechanism of that food effect must be well understood in order to translate that and to have confidence. And we are quite realistic here about bottom-up predictions. So we're aware that although drug-drug interaction modeling is having now regulatory impact with waiver of clinical studies, there is always that need to have some validation of the modeling with clinical data. And so we're also proposing that there does need to be some validation with clinical data before application of pure bottom-up modeling to predict food effects. And of course, as always, confidence will be increased by increasing the number of publications and ideally by some cross-industry efforts on model verification. And so that's really a lot of the motivation behind the current paper, which is now in press, and you can find it online at the moment in a preprint form. And yeah, it's been a great honor to work with Christoph from Janssen, Tanay and Tycho from Novartis, Philippos and Binfeng from Merck on that paper. And I encourage you to look out that paper for more details of the case studies presented today, as well as other things. And one other thing that we address in that paper is the issue of a consistent workflow and strategy. And so in the paper you will find this figure, and with this figure we try to come to some kind of consolidated strategy. So we start at the top of the figure with those criteria which we feel are supportive of the likelihood of a reliable simulation of food effect. So we've already mentioned BCS classes 1 and 2 as those where we have the high level of confidence. because usually we understand the mechanisms of the food effect for such molecules. Of course, we accept that when there's involvement in transporters and poorly permeable molecules, then we still have gaps in understanding. And so if we see non-linearities in PK, that can be a warning about reduced confidence. And of course, we mentioned that we would need some clinical data to perform model verification. So then we outline our strategy, which is in terms of building the first human model, making a first simulation using the default physiologies built into the software platform, and then potentially carrying out some model optimizations. At that point, we could then move on to making a prediction of the food effect, making a prediction for the non-tested prandial state. So usually that would be the fed state, assuming that the first studies were done in fasted state, but it could be different. And again, we would be able to verify the model using a clinical food effect data before then moving on to the later stages of development, where assuming that we have by that stage established a high level of confidence in the modeling, then we would expect to be able to leverage the model for predicting untested scenarios, for example, a different formulation such as I showed, potentially different types of meal as was shown by Tycho, and another example could be the timing of dosage with a meal. So that's what we have come up with. Of course, we outline also the importance of sensitivity analyses and virtual trials in this whole process. But we hope that this will be helpful to move us forward to more regulatory impact of PBPK modeling in this area. And so to close my part of the presentation, I'd just like to mention two other initiatives in which I am involved. The first one is the UNN Gap initiative. This is standing for European Network Initiative on Understanding Gastrointestinal Absorption Related Processes. And you can see from this slide that one of the working groups within this initiative is focusing on food-drug interactions. And so, there is a lot of top quality scientific input to this initiative, and we can expect improvements in vitro and in vivo understanding from this activity. And if you want to look into that more, please look up the web link there. And then finally, I'd like to mention an IQ working group, which has been active since the beginning of this year. It's actually a very active working group with regular WebExes taking place. Very good representation from 12 different pharma companies, with multiple representatives from each company, including both formulation and modeling expertise. It's been very ably led by Arian from AbbVie, and the goal of this working group is to assess performance of mechanistic absorption modeling for food effect following a consistent strategy, such as very similar to what has been outlined in our recent paper. And so we're putting together a large data set of compounds with which we will assess the performance, and we're expecting to write that up in a paper where we outline industry view on best practice, and hopefully moving forward this area through this collaboration. So thank you very much for attending this. Thanks a lot to the co-authors of the recent paper. And yeah, let's open up now for discussion.
Thanks, Neil. Thanks, Tycho, for the presentation. All attendees, if you have questions, please submit them via the question box on your GoToMeeting interface. We already have received quite a few of them, so we'll just go down the list. But please keep sending the questions. I will also add that in addition to Neil and Tycho, Christoph has also joined us as a panelist, as well as Viera Lukasova from Simulations Plus. So with that, the first question was around how confident are we when we rely on in vitro data to predict in vivo permeability? Tycho, this came during your presentation.
Yes, so I would submit that if you have a high permeability determined in KCO2 and in Pampa, then you probably have high confidence in your permeability assessment. But remember that the permeability can be assessed other ways other than just through the in vitro data, right? If you have a radio labeled data, then you can also determine what was the absorption of a given compound. So I think the confidence can be lowered in the actual input that goes into the model when you have low permeability, or when you have compounds that are extremely lipophilic, where you can have this bile acid complexation. So I guess in sort of summarizing this, when you have the low permeability, that's where you can have significant sensitivity, of course, first of all, but number two, it could be that the numbers could be not as high confidence as high permeability, right? If you have a compound, high KCO2, high Pampa, the chance that all of a sudden you have no permeability in a in vivo relevant system is actually low.
Thanks, Tycho.
Yeah, maybe if I can just add one point, Tycho. I think also the use of the preclinical data, for example, in the rat or dog or monkey, can also be used to support preclinically, the permeability of a molecule. So, are we able to predict the absorption phase well in those preclinical species?
Yes, absolutely.
Second question was around, I think Tycho briefly mentioned transporters. So the question was around transporters. How do you evaluate the effect of bile salts and food on inhibiting intestinal transporters? There are some reports, that some of these components can have interaction with drug transporters. Can anyone take that up?
Yeah, so I think we have basically a transporter group who specializes in this type of assessment, but to identify the risk that intestinal transporters could be involved, we usually find this out through that overall the permeability is moderate and can be impacted by transporter drug interaction at this stage. And then also in terms of biopharmaceutic properties, these are drugs that are usually BDDCS 4 drugs, where permeability is not just dependent perhaps on a transporter, but it's also region dependent. So in that case, we would do the transporter panel assessment, like say for BCRP, expressing cell line and adding inhibitor, for example.
Right, and I think we have acknowledged that where there is poor permeability, then the confidence level is dropping, and there's still a lot of work to be done there to translate in vitro to in vivo in terms of transporters.
I think in terms of modeling, we had one example in the past where the drug was a substrate of the the apical sodium transporter in the gut, and we found out that we can use GastroPlus to interpolate for certain type of clinical studies. So you may have a high dose study and a low dose study, and then you can interpolate, but the prediction there is probably a lower confidence, and especially when it comes to regulatory agencies, they're going to be more open-minded to predicting something for a dose group that's bracketed rather than to an extrapolation case.
Thanks. Next question. There is a question around lymphatic delivery. Food can trigger lymphatic transport, and how can that be accounted in a simulation? If any of you can comment. And I have some thoughts, but please, guys, go ahead first.
I do not have much experience in this area for lymphatic delivery.
I think that's very challenging. Of course, I mean, I don't think you can include that in GastroPlus at the moment, but Vera will correct me if I'm wrong there. But honestly, I haven't seen a lot of molecules where they are sufficiently lipid soluble and poorly solubilized in aqueous media that lymphatic is playing a major or expected to play a major role. There are some rules of thumb you can follow, I think, to estimate lymphatic absorption, but again, it's not an area where we would have high confidence.
Yeah, I agree. I would just add, I think, lymphatic transport is a post-absorptive event, so it's limited to a relatively small number of compounds that are extremely high lipophilic, and you have to have high first pass metabolism to see an effect. So I haven't observed many cases that lymphatic transport. I think there's a couple of publications out there, but I haven't seen it with other compounds that do not fall into these categories. Next question is, the examples focused on BCS class 1, 2. In the paper, we also mentioned 1, I think, in the title. How about BCS 3 and 4? Can either of you comment on BCS 3 and 4 compounds?
Yes, so I think for BCS 3 and 4, what we can basically do with confidence is to identify certain mechanisms which result in reduced exposure and presence of food. So I mentioned the one example where we know that we have to use a KCO2 system with biorelevant media to figure out if surfactants complex the drug, solubilize it, but complex it so the drug is not available to absorption. And then we also have some other assays where we can identify the risk of a negative food effect, for example, in the dog, but the magnitude of that reduction, I think, is more difficult to predict. So risk, yes, but magnitude, I think, is difficult.
Okay, thanks, Lego. So the next question is that most publications that have case studies focus on either retrospective confirmation of a clinical study, mostly phase 1 IND type of studies. Could you comment if you are aware or you have specific examples where the modeling was used to waive the BAB food effect study and was accepted by an agency?
Yeah, this is an interesting question, right? Because it's a big question. As you know, I think also Neil alluded to this. We are usually in a business model of global drug development. So that means that we have to not just satisfy what FDA is looking for, but what is for example, the Japanese authorities are looking for. So that means currently, for example, the PMDA in Japan still prefers us to conduct the food effect of the final marketed form. So that means for us that we can predict that something is bioequivalent, but at least at Novartis, we still often run the study to meet specific regulatory demands. So regarding the question, do we have an example where a food effect study was waived because of the modeling? I can tell you that we internally use, of course, this to choose compounds or formulations that do not have food effects. So we have been successful with this. But outright waivers to not run a food effect study are rare. You will know that the IVC guidance, for example, specifies that you have to run BABE considerations under the fasted state, and only in very few cases exceptions are made that you can run it in the fed state, which means that you have to demonstrate there's no food effect. So current guidances tell you what you have to do there. It is our understanding that ultimately regulatory agencies will ask for input so that they have more confidence on how to use PBPK models to waive and accept the studies. But again, examples to my knowledge are rare where we can not run a study because of modeling.
Yes, I would agree with that. But I'd like to, go back to our proposal from the recently accepted paper. We're proposing there that we verify the model with an initial clinical food effect study, but then that we leverage that, assuming that it's predictive and that we have confidence, that we leverage it to extrapolate to another formulation or to a different meal timing. And so I can share with you that with the alectinib example, we did a food effect study, and we then used the modeling to predict the likelihood of reduced exposure with when the dose was taken delayed from a meal by a certain time, and that was submitted as part of the filing. So yeah, I think our initial aim is to have impact on repeated food effect studies, and to refine the guidance with respect to formulation and timing effects.
Thanks. There are a couple of questions around precipitation and supersaturation precipitation, equilibrium versus kinetic solubility. There's 2 or 3 of them. So any comments around the handling of these and the effect of food and in the simulation of these parameters?
So we have some limited experience in this world because, as you know, there was a recommendation from the Orbito consortium about how to conduct biorelevant dissolution testing using a 2-stage dissolution method and using FASIF and FISIF media. This was published, I want to say, sometime in August last year, and I suggest that companies or individuals read the paper and take a look at it. I'm not on it, but I think it's a good paper. We have been using this recommended method, the 2-stage biorelevant dissolution method, to evaluate the dissolution media and then trying to gather impacts on the precipitation rate from the slope of the line. But I think the verdict there is probably still out.
Thanks, Diego. The next question is around water volumes. And I think this came during Neil's presentation around modifying the water volume in the simulation. And is it done like for all drugs, or that was a specific example? If you comment a little bit more on that. I think, Taiko, you also had something on the water volume. If both of you can comment on this.
Yeah. So I didn't go into the details of that. The example that I shared is quite an old one, and when we carried out the modeling at that time, the GastroPlus software did not incorporate the regional dependence of bile salts, and so our approach at that time was just to put in the biorelevant solubility on the main compound tab, and then of course that would be used throughout the intestine. And in view that bile salts by the time you get to the distal intestine are more or less completely reabsorbed, that is probably not physiologically correct. And so when we were preparing the paper, I went back to the model and used the recent version of GastroPlus to account for that. And indeed, once you do that, then the absorption from the distal, the colon is not too high. So probably that adjustment was compensating for the incorrect use of solubility throughout the GI tract. So that's a bit of a discussion of that particular example. But I think everyone will acknowledge there is some controversy over the amount of water volume in the GI tract at the moment, and indeed the most recent version of GastroPlus includes a dynamic fluid volumes description.
Yes, if I can just add to that, because we know that others have also modified the colonic and intestinal water volume. It is important to remember that only certain compounds are going to be sensitive to that, right? I mean, I'm just hopefully stating the obvious, but you would need to have a high permeability compound in the colon for it to make a difference. So if you have a compound with moderate permeability, probably it's not going to make much difference what water volume you have. So I guess my feedback, and perhaps provoking some thought for this user group, is that let's look at compounds which we know have high permeability, and then look, is a lower number warranted across compounds. So if somebody tells me, well, I didn't have to change it and it worked for me, well, that's fine, but I need to know that the permeability was high in that case, because only high permeability compound will probably see a sensitivity there.
Thanks, Taiko and Neil. There is a few questions around animal data. I mean, there are different aspects of them, but just to group them together, using animal data to predict the human PK, what's the industry experience with this, and how do you set some of the settings in the models when you move from an animal to a human, you're simulating, let's say, food effect?
Yes, so I think animal, particularly dog studies, are still being conducted by some companies, including, if anybody's on the line, from BMS. they have done a good job publishing the dog as a food effect model. What I would submit is that we use animal data in the integration of food effect models multiple ways, because if you look from a regulatory point of view, it is most preferable to have human mass balance data from a radio-labeled study and intravenous data to be included in a food effect model. But this is not practical because some compounds, it will take for a long time or never any intravenous data is generated. So then in those cases, you need to use animal data to scale the disposition parameters, and you can do that with multiple methods. One of the papers I showed there are other papers as well, and I believe the Orbital Consortium is also going to talk about this bottom-up approach using animal data, but more specifically to food effect alone. We have been using under limited cases still the dog as a model, but we are not running nearly as many food effect studies in the dog as we used to, because again, we're using GastroPlus to anticipate the risk. The compound that I talked to you about, the compound N, we did not run any animal studies.
Yes, I would agree with you, Tycho. We are now hardly doing dog food effect studies, whereas 10 years ago we were doing those fairly frequently, and now we tend to rely more on the biorelevant tools and the PBPK modeling. I wouldn't want to rule out completely the value of the preclinical model, but one of the issues is how well we understand those preclinical models in terms of physiological inputs, and I think it's fair to say we have much better understanding of the human situation than we do the preclinical models. At one stage, we invested some efforts to better characterize those animal models, but I think we're really tendency is now to go more for in vitro biorelevant tools and the PBPK modeling relevant for human.
Great. There is also a few questions around dissolution related aspects. I'll just combine them again to a long question. So can you generally comment around reliable in vitro dissolution tools to predict food effect for different meal types, since the presentations gave some of the low moderate fat simulations too, and can you also comment generally speaking biorelevant media, use of them for the simulations, etc. Can you have your general thoughts on the topic?
So I think I'll start out with a higher level of comment. Philippos, as you know, we have had this MCERSI FDA workshop last year, and it's my understanding from regulatory agencies that just because you're using biorelevant media, if you wanted to set up an IVIVC or sort of a prediction, just because you're using biorelevant media, that alone does not guarantee you a regulatory acceptance. So the concept of using biorelevant media is certainly appreciated by the regulatory agencies, but it's not sufficient. So I think one thing you can anticipate, there will be two publications coming out from that FDA MCERSI workshop, probably in the very near future. And again, the paper from the Orbital Consortium, I think, is probably state of the art.
Okay, thanks. All right. Very interesting question. Any experience or are you aware of any ongoing efforts to use PBPK modeling to predict the effect of alcohol on PK?
We do not have such a recent example at Novartis. It's probably relevant to dose dumping, but perhaps you, Neil, or Vera will have, or Christoph has an example.
I remember, I didn't work on it myself, but I remember we did have a few years ago a compound where we carried out some in vitro solubility and dissolution measurements with alcohol in order to make a prediction using PBPK for the effect of alcohol. I'm not sure, it wasn't my project, so I'm not sure if that prediction was ever verified. I'm not sure, but you know, obviously it's something that is feasible.
Okay, thanks. Let's see. A few more questions. We only have like 10 minutes left. The examples shown are immediate release formulations. What's the experience with sustained release formulations? And do we know if the models take into account transit times for sustained release systems?
Yeah, so I think this is an interesting concept, because you will know this is exactly the type of discussion that we had sometimes with regulatory agencies, because the argument is food effect is difficult to predict, and then you have a BCS class 1 compound which was converted into a modified release formulation, and then of course you can see food effects because you have now altered the properties of the drug, and the fraction absorbed may or may not be complete in the fasted state, such that you would get a positive food effect for compounds such as propranolol or metropolol. In this user group here, we did not include any of these compounds. My recommendation would be for us to look at data sets where we have a BCS class 1 compound and where somebody made a modified release formulation, and then we can see if we can accurately and with confidence predict this. So this would be sort of a second generation, but we have not had many modified release formulations where we needed to evaluate the food effect.
Yeah, I think there are some examples which have been published on using dissolution data for modified release formulations and seeing the food effect change. I think in the paper that I mentioned in 2008, we included one such example, and I think there are, I seem to recall some others groups have published on similar attempts.
Thanks. This is a question specific for Neil. Neil, the question is, in your example, the food effect was predicted well for Cmax and AUC, but Tmax was not as good. Can you comment on Tmax predictions for food effect?
Well, yeah. So that's true when you look at the mean profile. When you look at the individual data, I remember for that example we had a lot of variation in Tmax with a lag time type of effect. So yeah, that was quite a variable Tmax for that particular molecule, and maybe just the point estimate wasn't giving the full picture there. I think generally you you see a reasonable prediction of Tmax with the delayed Tmax with food with BCS II molecules. That's generally my experience.
Mm-hmm.
Yeah, this is a good segue. There's an associated question around addressing the variability in gastric emptying and retention time, and how does PBPK help predict it? So maybe you can just comment a little bit on the variability of these settings for the models.
You see, if you want to look at variability in terms of the physiology, well-behaved compounds are best suited to evaluate that. So, for example, when you have a compound which is a BCS class 1, it lends itself to evaluate what happens, for example, for gastric emptying, because the other parameters may not be rate limiting. And so I think this is where within the user group community, I like to know, based on the recommendations which we have had for the gastrointestinal pH, right? The numbers we put in there actually were based also on some orbital findings, where a study was conducted by Patrick Augustine and others. They looked at the gastrointestinal pH and the variability. So I would like to know from the user group community, if we have 100% CV on gastric pH, is that going to be something which other labs can corroborate with other compounds? We have had very good experiences with predicting proton pump inhibitor interactions, actually the lack of proton pump inhibitor interactions, using a high variability along in a population of 100%. And so then the recommendations for how to include variability should also speak about what can we do to include the metabolism there, right? So we can put the POP-PK estimate if we want to use a simpler approach, but the platform questions that are basically being raised here around variability is really something which the community needs to solve if we want to have a buy-in from regulatory agencies.
Yes, I agree, and I think it's good to highlight that what we need is more characterization of the variability in different populations. I think that's an issue which this UNGAP effort, which I mentioned, is going to be looking into. And I think also from the Orbito effort, there was quite some nice advances within that consortium. So I think you're right that that's something where we need good